Hello everyone, and now we're going to be talking about blood vessels. So, to begin with, let's talk about the names that we use for these blood vessels. And if we started at the heart, the order in which we usually encounter blood vessels is heart goes to arteries, to arterioles, to capillaries, to venules, to veins, which lead back to the heart. As we mentioned in a previous lecture, sometimes people make the mistake of thinking arteries, that means oxygenated blood. And that is true for the systemic circuit, but for the pulmonary circuit it's the other way. In the pulmonary circuit, the arteries are carrying the deoxygenated blood out to the lungs where it'll pick up oxygen, and the veins are carrying the oxygenated blood back. But this is the typical pattern. There are a few places in the body where we get something a little different. Notably, you get places where you get heart, arteries, arterioles, capillaries, and then rather than straight to venules, usually what you'll get is another set of arteries or arterioles, and then another set of capillaries, and then the venules and the veins. We call those portal systems, where there's more than one capillary bed in series. You find those in the kidneys, around the hypothalamus, and a few other places. And we're not going to pay a whole lot of attention to them right now, but I am going to talk about how each of these vessels is constructed and kind of how that helps it serve its purpose. So just to summarize those purposes, let me quickly erase and say arteries. The main purpose of arteries is to actually carry highly pressurized blood away from the heart, especially in the systemic circuit. They're under very high pressure. So these have to apply pressure and also withstand high pressure. Arterioles are mostly about controlling blood flow. Capillaries, we're going to pay special attention to capillaries later, are about exchanging materials between the blood and the ECF. Or in the lungs, it's barely even ECF. It's almost straight with the air. Then venules and veins are about bringing blood back to the heart. And in the case of veins, they have some anatomical features like the valves, which help them do that. Veins are also important in that they can act to sort of accumulate blood. So they're a way of diverting blood away from the arteries and holding it there for a little while, letting it slowly get back to the heart, in case you need to get blood out of the arteries to, say, lower arterial pressure. Or you can constrict the veins, push that blood back into the arteries, and raise arterial pressure. We talked about that a little bit last time when we talked about blood distribution. So, let's start by talking about each individual kind of blood vessel. So, erase, and let's talk arteries. Actually, before we even do that, let's talk about something that's in common with all of these blood vessels. That is the idea of endothelium and the basal lamina. So on the inside of every blood vessel, you will have a thin layer of cells So this is one cell, very thin. If we draw this in sort of pseudo 3 dimensions, you could see that this is one cell forming sort of a tile on the wall. This would be a very small vessel. This would be a capillary. And outside of those, there is a network of protein filaments, shown here in pink, called the basal lamina. Sometimes basement membrane is also used for that. So this is endothelium. and that's basal lamina. Basal lamina consists mostly of collagen, which is a kind of a fibrous protein, and some level of elastin, which is a stretchy protein, sort of like rubber. The more elastin you have, the springier the membrane is, the more easily it will stretch out. Some vessels, like arteries, have a great deal of elastin. Some, like venules, have very little. So, keeping that in mind, let's now talk about the structure of individual blood vessels. So, let's go with arteries first. Now, I'm not going to draw arteries down at that very small scale. We're going to draw them larger because many arteries are quite big. But arteries will have the same set of structures. One of them, the innermost layer, is the endothelium and a basal lamina and I'm going to add on to that basal lamina. So, in addition to that basal lamina, I'm going to draw in a little bit of extra, here in green, elastic tissue. Extra elastin in a thick layer on top of there. So here we have, this is endothelium, basal lamina, and arteries have a special extra thick layer of elastic tissue. It's really just an extension of the basal lamina. Those together form what we call the tunica intima, the innermost layer of the wall of an artery. The intima is just this, it's a fairly thin layer. It wouldn't look like much under a microscope, but it forms the layer that the blood sees. Now notice that basal lamina has a lot of collagen in it, and you know that when blood encounters collagen we get blood clots. But that's kind of what the endothelium is for. That seals the collagen away. Those endothelial cells, as we talked about in coagulation, also secrete that platelet lullaby that helps keep the platelets dormant and inactive. So that's the tunica intima. Now I'm going to erase this so we have more room to draw the rest. So just make sure you've written it down. The next layer in an artery is a fairly thick layer of smooth muscle. In arteries that has some elastic tissue in it too, but I'm not going to draw that specifically. That's smooth muscle, which we call the tunica media. Now, in arteries, that smooth muscle is there for a good reason. Number one, it adds to the strength of the blood vessel, helps it to resist the very high pressure from the blood that's pushing out on that. But number two, that muscle can constrict and apply pressure to blood in the arteries. And here's why you might want to do that. During the heartbeat, when the heart ventricle is contracting, that applies pressure to the blood, which pushes it on through the vessels. We've mentioned that. But what about when the heart is in diastole? What about when the ventricle relaxes? Then the heart is exerting no pressure on the arterial blood, and if there's no pressure, it's not going to move. This is where this tunica media and this extra elastic tissue come in handy. During ventricular systole, when the ventricles are pushing high-pressure blood into this artery, that high pressure pushes out the walls of the artery, stretching them, stretching all that elastic and smooth muscle. And then, when the ventricle relaxes, that elastic tissue and smooth muscle springs back, what we call arterial recoil. That recoil applies pressure. It's almost as if you blow up a balloon and it stretches it out, and then as you stop blowing, the latex of the balloon springs back and applies extra pressure to that air. That keeps the blood moving even when the heart isn't pumping. So that's actually one of the ways that arteries can apply extra pressure to blood to keep it moving. That also affects arterial pressure. And then outside that smooth muscle, our last layer is a sort of a sheath. Oh, that's a terrible drawing. We're trying to make that slightly more even. Kind of a tough sheath of mostly collagen, some elastin. That's the tunica externa, which is tough collagen and some elastin. That acts as a protective layer on the outside of the artery. It shields it from anything that might try to impact the artery from outside. It protects it from any sort of cutting or anything like that. It also acts as extra strength to help keep the artery together since it's under so much pressure. Some clinical considerations there. If you have damage to this tunica externa, say you had a damage in this area, you can end up with a situation where the pressure in the artery is enough to sort of cause it to push outward. Like so, to bulge out. And we call that an aneurysm. That'll have some smooth muscle coming with it. That's a very dangerous situation. When you have an aneurysm, a swelling in the wall of the artery, that's much more vulnerable. And if that breaks, we now have a leak in an artery. If that's a major artery, this can be very quickly fatal. A blowout in an artery, it's like a blowout in a tire. You suddenly lose a lot of the flow and all of that blood is now coming out. Not a good situation. So we want to keep our tunica externa in good shape if we can. All right. Now, one last thing about arteries. The outer diameter of arteries can be quite large. The largest arteries are a centimeter or two across. They're big. But the inner diameter of the artery, the inner space, is actually quite small. Even in a big artery, that's just a few millimeters across. So the walls of arteries are very thick, very strong. Okay, now let's go on to the next one, arterioles. Arterioles, I should write that. Arterioles are smaller. They have a tunica intima, which is the endothelium and its basal lamina. Not a lot of elastic tissue, it's a little bit of elastic tissue in that basal lamina. And then they have a tunica media of some smooth muscle. So they've got the tunica intima and the media. They don't really have a tunica externa. They don't have that tough outer sheath. They have a decent amount of smooth muscle, and the main reason for that is that arterioles are about controlling where the blood goes. So you've got an artery supplying blood to, say, your whole arm, and then splitting off into smaller arteries, and then those smaller arteries split into smaller arteries, but eventually they get down to the point of where they are arterioles. And an arteriole supplies one particular region of some tissue, like one part of a muscle. Those arterioles can do a lot of constricting or dilating. So if you've got two arterioles, let me set these down, and this arteriole constricts and this one dilates, the blood is going to tend to go to the dilated one. And then if we switched it, the blood's going to tend to go that way. So these are almost like valves controlling which way blood is going to go. This is also important. If you look at all the arterioles together, that's kind of what determines that whole idea of peripheral resistance that we talked about when we talked about blood pressure. That, remember, arterial pressure depended on cardiac output, how much blood you put into the arteries, and peripheral resistance, how hard it is to get blood out of the arteries. If all of your arterioles dilate, then it's easy to get blood out of the arteries, and arterial pressure will tend to go down as blood drains away. Likewise, if all your arterioles constrict a little, it's like tightening the drain on a bathtub. The water will take longer to get out. It's harder for blood to get out of the artery, so pressure builds up. There is some evidence that in many cases, hypertension, high blood pressure, comes from having many of the arterioles in the body just being a little too tight most of the time, thus raising your peripheral resistance and making it harder to get blood out of the arteries, leading to higher blood pressure in the arteries. But their normal function is to regulate where the blood is going by dilating or constricting based on local need. We talked a little bit about blood supply to tissue being based on local messengers like the sensitivity to oxygen. The arterioles are the things that are doing that sensitivity, that are responding to the oxygen. Okay. Now the next one is capillaries. We're only going to talk very briefly about those here because once we're done with the blood vessels, I'm going to go back and talk about capillaries in a lot more detail. But capillaries, first of all, they're tiny. And they've just got an endothelium and a basal lamina. And that's it. No muscle, no tunica externa, just endothelium and basal lamina. In terms of size, capillaries are typically in the range of less than 10 microns across. Now remember, a red blood cell is in the range of 6 to 8 microns across. So when we get to capillaries later, you'll see kind of this in rough proportion. But capillaries are tiny and they have very, very thin walls, which they need in order to be able to have materials diffuse from inside to out or out to in, so they can exchange stuff with the extracellular fluid or the air. So we'll get back to capillaries. Venules are boring. A venule is really just a bigger capillary with its endothelium and basal lamina, and then a little bit of tunica externa. Just a little bit of tough tissue out here. Not much muscle to speak of. Think of it like a slightly larger armored capillary. Not really very interesting. So let's get on to the last ones, which are kind of interesting. Veins. Veins have the same layers as arteries, but in different thicknesses. So in a vein, we will have an interior endothelium with its basal lamina. The basal lamina in a vein does contain elastin, but not as much as in an artery. They aren't as stretchy. So that's the tunica intima in a vein. They will have a layer of smooth muscle, tunica media. And they will have a tunica externa, a tough layer of collagen with some elastin in it. So they'll have an intima, media, and externa, but notice the difference from the artery. They are comparable in terms of exterior size. The big veins and the big arteries have not too dissimilar diameters externally, but internally, veins are much, much bigger. Their walls are thinner. That's one of the reasons why blood can tend to sort of pool in veins. They can be thinner because the pressure in the veins is much lower than in the arteries. By the time you get to the veins, most of the pressure has been worn away with friction with the walls of the vessels. So there's very little pressure left at this point. That actually leads to one of the interesting things about veins. If we imagine a vein here coming up toward the heart from, say, the feet, this is carrying blood from the lower body up toward the heart. Now, we would normally need pressure to push that blood up, but remember there's very little pressure in the veins. In fact, there's usually not enough to effectively push against gravity. So how are we going to get blood back up to the heart from the feet? These veins have one-way valves in them. Flaps that open toward the heart, but then will close if blood tries to flow back. So if you get blood past this valve, and then you lose any pressure, it falls back, but it only falls back against that valve. And then if you can get it up past that one, it falls back. It's a little like putting your foot on the step of a ladder and then pushing up. You've got a place to stand. It's kind of like that. One thing in veins that helps us move blood around is the fact that veins tend to be located near things like skeletal muscle. So let's imagine that we've got a couple of skeletal muscles around this vein. When those muscles contract, the muscles bulge and push inward on that vein, which applies pressure to the blood in here. Now the blood can't go down because of that valve, so when those muscles apply pressure, it pushes blood up through that valve. So it supplies a pumping action. Then when the muscles relax, that blood from beneath kind of gets sucked up into that vein, and then they contract again and the muscle goes up, so the blood goes up. So if I want to increase the amount of venous blood getting back to my heart, the more I move my muscles, the more that skeletal muscle pump helps to push that blood back to my heart. And the more venous return I get, so the bigger my EDV will be, the bigger my stroke volume, the bigger my cardiac output. Kind of handy. There's also an action of a respiratory pump that the inferior vena cava, as it comes up through my abdomen and thorax, as I breathe, the shifting of pressure with my diaphragm downward and then upward changes the pressure in my thoracic and abdominal cavities kind of back and forth. That also helps to pump blood upward from my lower body. So all of that helps to get blood through the veins, even though it's normally under very low pressure. Okay, that's blood vessels. That's the major blood vessels. Now, we want to pay special attention to the capillaries, because that's where the really interesting stuff happens in terms of physiology. So let's take a moment to get into capillaries. Okay, these are the tiny blood vessels. So let me draw a capillary in close-up cross-section here, sort of like we did before. So here's our endothelial cells. And our basal lamina around it. Now, as we mentioned, capillaries are usually less than 10 microns across. Usually we're talking 5 to 10 micrometers across. So we'll say 5 to 10 micrometers, millionths of a meter. On that scale, a red blood cell would be about that big. So red blood cells pass through these capillaries in effectively single file, maybe two if it's a larger capillary. So these are tiny, tiny vessels, and there's a lot of them. The total, I think I read somewhere that if you took all of the capillaries if you took all of your capillaries out of your body and put them outside of you end to end, you would die. But before you died, you would find that there were something like 25,000 miles of capillaries. There's actually a lot more capillaries than you have blood to fill them, which is kind of interesting. At any given time, many of your capillaries don't have blood in them. Interesting to think about. Anyway, back to this. Now, the walls of these are thin enough that material can diffuse from the blood plasma to the extracellular fluid or back very easily. And we're going to talk in a moment about how stuff moves back and forth across there. In fact, let's talk about that now, at least for here. So one thing that we should pay attention to is here where these endothelial cells join. These, in most capillaries, join in what we sometimes call a leaky junction. And at leaky junctions, so if we draw that leaky junction in close-up, the seal between the two cells isn't quite perfect, which means some materials can move from inside to outside or outside to inside without having to go through the cell at all. Only very small things, though. So at leaky junctions, water and ions can pass in and out of capillaries. Anything bigger than that, glucose, amino acids, proteins, cells, all of that stuff, those can't go through leaky junctions. Those are going to have to go through the cell itself. And cells have ways of letting that through. They have transport vesicles, they may have channels or carriers. They will allow things through in many cases. But water and ions pass between the cells in addition to through the cells. All right, so that's part of the basic idea of a capillary. Now let's talk about types of capillaries. So what I've drawn here with the leaky junction is the most common kind of capillary. If we kind of extend this out again into that pseudo 3D drawing, and draw on my basal lamina, then what I've got is what we call a continuous capillary. Most of the capillaries in your body are continuous capillaries. Continuous, they're called continuous because there's no gap in the wall. It's just endothelial cells. They have that leaky junction, so water and ions can squeeze in and out, but everything else has to go through the cells if it's going to pass. So that's why they're called continuous. Most of your capillaries are continuous capillaries. If you're not sure whether something is continuous or not, it probably is. There is a special case of these. So this is most of your capillaries. In the central nervous system, the leaky junctions become tight junctions. Tight junctions are sealed up even against water and ions. So in the central nervous system, in the brain and spinal cord, these leaky junctions seal up, and even water and ions have to be allowed through the cell. The central nervous system takes very, very close care about what gets into and out of it, because the brain and spinal cord are delicate tissue. So, by sealing up those leaky junctions, and then also some of the glia in the brain, the astrocytes, sort of put down pseudopods that partially cover these capillaries and provide another layer sort of filtration there. That actually helps to make sure that only things that we really want to move get out of the blood and into the brain. When you've heard people talk about the blood-brain barrier, that's what we're talking about. Both changes in the endothelial cells and how they're joined, and the astrocytes in the brain and the spinal cord create a sealed capillary where we control much more carefully what gets in and out. That's good most of the time. It makes it harder for disease organisms and some toxins to get to brain tissue. It's bad when we want to deliver pharmaceuticals to the brain. A lot of drugs are difficult to get into brain tissue just because they don't pass through the barrier. So anyway, that's a continuous capillary, and the special case of a continuous capillary in the brain and spinal cord. Now, the second type of capillary. Let's see. We'll redraw this. We'll erase all of this. These are a little hard to draw, but I'll do my best. So what I'm trying and largely failing to draw here is the idea of what we call a fenestrated capillary. Fenestra means window. I think it's Latin. A fenestrated capillary has little pores, little holes in the endothelial cells, almost like a colander. These pores are about 60 to 80 nanometers across. To give you a sense of scale, a nanometer is about 10 hydrogen atoms. So these are a couple of hundred hydrogen atoms across. They are still kind of under this basal lamina, which acts as an additional filter. But having these pores means that materials bigger than water and ions can move through this. Between the pores and the basal lamina on top of them, this allows water, ions, and small organic molecules like glucose, amino acids, most metabolic wastes, anything smaller than a protein can move in and out of this blood vessel without having to go through the cells. So we can't even control that movement at this point. It's just holes that those things can go through. So these fenestrated capillaries have a lot less control over what goes in and out. But on the other hand, larger things can move in and out of them easily. And if you think about it, there's a few places in the body where we would definitely want that ability. If we want to easily be able to get organic materials, like organic wastes, out of the blood, where in the body would we want that? Definitely in the kidneys. That's how we're going to get a lot of these organic wastes out of the blood. Now, where in the body would you want to get materials into the blood? If you're bringing, say, nutrients in from outside the body, where would you want to be able to make sure you got them into the blood easily? That would be the intestines. And one other place where you find these, in areas where you want to be able to put some other materials into the blood, specifically the endocrine glands. In many cases, you'll find fenestrated capillaries there. Places where you might want to get a hormone and put it into the blood easily. So, fenestrated capillaries are specialized in a few areas. They're good at moving slightly larger things into and out of the blood easily, rather than having to transport them through the cell very deliberately. Okay, so that covers fenestrated capillaries. There's one more type to talk about. The last type we call sinusoidal. Sinusoidal capillaries are weird. First of all, they're much bigger than regular capillaries. These are on the scale of, I think we're talking maybe 40 microns across. They're something like 5 times the size of a regular capillary. I'll try to get that across here. Those endothelial cells are way too big. The other really interesting thing about these capillaries is that they have giant holes in their walls. So these cells don't even really seal together at all. They've got these enormous gaps in the wall. Gaps big enough that things like whole red blood cells can get into and out of the blood here. Now, why on earth would I want a blood vessel that doesn't even hold in blood cells? What's the point of a sinusoidal capillary? Where would I want that? Well, there's 3 places we can think of pretty easily. One, we have one organ in which the cells of the body not only need to put a lot of proteins into the blood. Remember, proteins are too big to fit through fenestrations. So they would need big gaps. So we want to put proteins into the blood, and we also want to be able to access and modify just about anything in the blood. That's the liver. So the liver is full of these sinusoidal capillaries, which are barely capillaries at all. They're more just sort of suggestions of where a capillary might be. And the hepatocytes, the liver cells that live outside of these capillaries, have direct access to everything in the blood. Another place, red blood cells not only can come out of these, they can go in. So if we think about where do we make red blood cells, that's somewhere where we would need to be able to put cells into the blood easily, and white blood cells for that matter. And that's the bone marrow. It's where we make red blood cells and some white blood cells. And finally, a place where we want to be able to get rid of red blood cells, the place where we get them out when they're dying, the spleen. These 3 areas tend to have sinusoidal capillaries, which allows them access to everything in the blood, including proteins and the cells themselves. So that pretty much covers blood vessels. In the very last part of this lecture series, we're going to go back to capillaries and talk about how it is that water moves into or out of those capillaries and how we know which way it's going to go.